Assalamu alaikum wa rahmatullahi wa barakatuh. Respected teachers, worthy scholars, dear friends, I am Mohammad Mosa Mubarak here to present my dissertation thesis entitled Epigenetic Modulation of Lovastatin Biosynthesis in Aspergillus terrus and endophyte of Glycyrrhiza glabra. Completed under the joint supervision of Dr. Sayyid Riyadul Hassan and Dr. Hamid Wali. In our present times, when we are faced with the challenge of drug resistance, new disease outbreaks, and inefficient diagnosis, our hope lies with the microorganisms, which are the foundations of our biosphere and which have been contributing from past 100 years to the industrial development. More than 60% of anti-cancer drugs, 70% of antimicrobial drugs currently in clinical use have been derived from microbial sources. However, there needs to be an exclusive exploration, as there are only 1% of bacteria and 5% of fungi that have been characterized. Bioprospecting of these microorganisms involves exploring these microorganisms in their environments and habitats, be it extreme ones like ocean beds, geothermal vents, cold deserts, and medicinal plants. One of the endeavors of bioprospecting is the endophyte. Endophytes are microorganisms that reside within the living tissue of the host plant without causing any sorts of symptoms of disease. Yes? Okay. This? The endophyte in our study is Aspergillus terrus, which was isolated as an endophyte from Glycyrrhiza glabra, also known as licorice or esteemed. Aspergillus terrus is a filamentous fungus that can be found in soil isolated from plant species. It exhibits a brownish color, possesses conidial heads that are columnar biseriate. Most of these drugs that I was talking about are secondary metabolites. These are the compounds that are secondary to use to the organisms secreting them. Secondary metabolites produced by endophytes are constant in very considerable in the drug industry. Our endophyte Aspergillus terrus is also known to produce a variety of secondary metabolites that have industrial use. We have ecotonic acid, cis-ecotonic acid in the paper and the textile industry. We have Lovastatin, the drug under study here, which is a cholesterol-lowering drug. Lovastatin is a member of the statin group of compounds that specifically inhibits 3-hydroxy-3-methyl-glutyl coenzyme reductase in a long biosynthetic pathway, which later leads to the production of cholesterol. The biosynthesis of lovastatin in Aspergillus terrus is controlled by this huge cluster of genes known as the type 1 polyketide synthase. Among these genes, lovastatin B and lovastatin F synthesize nanoketide synthase and diketide synthase, which are key enzymes for the biosynthesis of lovastatin. Besides these two genes, there are other genes and other open reading frames that regulate the synthesis of lovastatin in Aspergillus terrus. Okay, since Aspergillus terrus is a fungus and fungi are eukaryotic organisms, these are transcriptionally controlled. Their genome is transcriptionally controlled by epigenetic regulation such as histone acetylation and DNA methylation. In our study, we have used 5-azacitidine, which is an epigenetic modulator that interacts with the enzyme DNA methyltransferase to bring about its degradation and cause DNA hypomethylation, which later on leads to chromatin restructuring and activation of gene clusters. Now that we are all on board the theoretical part of our study, let's move on to objectives. Our objective in this study was first to develop the epigenetic variants of Aspergillus terrus. After this, we were interested in studying the effect of Aspergillus terrus vis-a-vis lovastatin production, growth rate, secondary metabolome of these variants, the changes, if any, in the morphology of these variants, and the overall biochemical profile of these variants. We also ventured through the expression of selected genes involved in lovastatin biosynthesis post-azacitidine treatment. Now the methodology, our methodology involved first subculturing this fungal endophyte Aspergillus terrus since it was previously isolated from Glycyrrhiza glabra and characterized by ITS sequencing. We subcultured it on PDA plates, potato dextrose agar, and then maintained it at 28 degrees Celsius. Yes? Sir, this is the formatting error. I had made my presentation in Word 2007. This is Word 2010. Hence, I think this should be admissible. Okay. After the subculturing, we went for the azacitidine treatment. We gave 3 different concentrations of azacitidine. We have 50 micromolar, 10 micromolar, and 5 micromolar. And as such, we developed 4 different epigenetic variants. We had an indigenous strain, GG17, which was original Aspergillus series. Then we had A50, which was an organism exposed to 50 micromolar concentration of azacitidine and grown without any subsequent exposure to aza. Then A10, which had an exposure of 10 micromolars, then 2 subsequent exposures. A5 with 5 micromolar exposure and 2 subsequent exposures. In short, we had 4 different variants of Aspergillus series throughout our study. Since we were interested in analyzing the growth of our different epigenetic variants, we did so by developing a growth curve. This was achieved by first inoculating each of these funguses onto a PDA plate, and these plates were then grown for different time intervals. 5 yes. Sir? Sir, I can't understand. Yes. Huh? Okay, this is the protocol, like inoculation.
No, it is not a protocol, it is a grammatical mistake.
Okay,
grown for time interval. Grown.
Okay, in past tense. Grown for time intervals, these are 5 days, 7 days, 9 days, 12 days, and 15 days. At each time interval, the mycelia was scratched off using microtip, and this mycelia was dissolved in NSS. This mycelial solution was then serially diluted, and at a particular dilution, the inoculum was taken and it was spread on a PDA plate. This plate again was grown at 28 degrees Celsius for 3 days. After 3 days, spores were counted. And the counting of these spores was put into an expression which gave us the CFU per ml, colony-forming units per ml, and that data at each time interval was then formulated into a growth curve. Since we were also interested in understanding whether there were any morphological scenes at microscopic level, we did the light microscopy using lactophenol cotton blue dye and got some images. Since the most important part of our study was the fermentation part, where we were interested in understanding the changes in lovastatin production by these epigenetic variants developed, for this purpose, we first grew the cultures in 500 ml PDB for 15 days. Afterwards, the cultures were extracted, the second metabolites were extracted using the National Cancer Institute's protocol. Ethyl acetate was used as organic solvent. Then this organic solvent was evaporated and solid extracts were obtained. These extracts obtained, dry extracts, were then analyzed by chromatographical techniques of PLC and HPLC to quantitatively and qualitatively confirm and quantify the lovastatin. Since we were also interested in studying some of the genes involved in lovastatin biosynthesis to see if there was any change in the expression of these genes. This was done by first growing these different variants for 15 days. Afterwards, purifying the mycelia. The mycelia was then subjected to isolation of RNA by the Trizol method. Then, after the purification of RNA, we quantified it using the nanodrop method, as well as through the agrosgel electrophoresis. We checked for interaction of this RNA. After that, we subjected like 2 micrograms of RNA to cDNA synthesis. After cDNA synthesis, we confirmed cDNA synthesis by amplifying it using beta-actin, the housekeeping gene. After confirmation, we went for RT-PCR, wherein we used primers for our selected genes and saw the expression in different epigenetic variants. Okay, since our since our organism is actually a fungus, an endophyte, and it provides growth promotion and biocontrol in plants, we are also interested in studying the biochemical profile of these epigenetic variants developed. Now the results. After the treatment of azacitidine. Here are the 4 images of the 4 strains of the 4 epigenetic variants that were developed. Figure A is the indigenous strain. It is not clear here, but it has brown pigmentation and has a particular growing pattern of the plate. In figure B, we still have the brown pigmentation. It is A5 with minimum exposure to azacitidine. Here in A10, we have green tufts here developing, and A50, we have the entire pigmentation being changed to green. The growth curve analysis gave us this data, which was formulated into this growth curve. Here we have each epigenetic variant, each strain color coded. We have the fungus with the fastest growth rate here, GG17, with no exposure to azacitidine, having the highest growth rate. Then we have dark red color here, this with squares. This is A5 with minimum exposure to azacitidine. It again has comparatively higher growth rate. Then we have A10 with 10 micromolars of azacitidine exposure. Here again, its growth rate is diminishing, and the highest exposure of azacitidine, which was given to A50, it has the least growth rate. Each of these epigenetic variants reached their stationary phase by 9 days.
Musa, these are not epigenetic variants.
Sir,
I said to you before.
Sir, you said these are not strains.
These are variants. These are you have given just different. These are not epigenetic variants.
Okay. So what should I call it?
Just same strain with different concentration, exposed to different concentrations.
So, but the epigenetic with exposure to azacitidine, the epigenetic exchange.
Yes, but these are not epigenetic variants.
Sir, I have. Okay, sir.
What should I call it? Not strain has been treated with different concentrations.
Okay.
Same strain treated with different.
So I should use the word strain instead of variant. So, these are the results of light microscopy. Here we have the conidia of Aspergillus terrus exposed to 50 micromolars, and here is the indigenous strain. The difference is of microscopy, although the morphology is the same. They are found to be septate, hairy with acute angle branching, and the pre-light branching. Okay. These are the representations of the extracts obtained after the fermentation processes. Different colors of the secondary metabolite solution indicate different secondary metabolite concentration. After drying, we obtain different weights. We have GG17 with minimum weight of minimum concentration of secondary metabolites. A50.
There is one question, but we will take later on.
Okay, sir.
You are showing that the GG17 is having maximum growth.
Yes.
In presence of azacitidine.
Yes.
So when there is
It is
in presence of that.
No, GG17 has no exposure. That's the control. That's the indigenous strain. It is normal. Yes.
Obviously, it is normal.
Yes.
It is having maximum growth.
Yes.
It is normal. It is having maximum growth.
Yes.
Is the red one?
Yes.
You are showing the red one or the blue one?
It's the blue one.
The normal one. So you are giving an azacitidine. You are producing these secondary metabolites.
Yes.
Why in first case when it is having more growth at that time?
Yes.
Why it is having less these secondary metabolites?
There are multiple reasons for that. Since number one, there is the exposure by azacitidine, which has.
So there is no exposure.
Sir, which? I don't understand your question. Okay, let me. We have the no exposure GG17. It has minimum concentration.
Why? Why?
Okay. It will be answered by this. Huh? We have the maximum concentration of at A50. Okay, sir. At A50 we have maximum concentration. So it is there is change in epigenetics. New cryptic pathways are open. More and more secondary metabolites.
My question is second thing.
Okay.
It has been established, well established that when there is more biomass, obviously more will be secondary metabolites. Because secondary metabolites these are contributing towards.
Yes, I don't see here.
But it is maximum growth, but you are showing little secondary metabolites.
So
But you know, the thing over here is that when he is using ESA,
Then maybe he has
he is inducing the cells.
After discuss.
He is inducing the cells to make more
Actually, my question
secondary.
not that thing. Here's this saying only about that thing. That is one thing. Lovastatin. Lovastatin comes to.
No. This is all of the secondary metabolite. This is all of that.
So Lovastatin is speaking about that. So that is not the whole extract. There should be compensatory weightage of the whole extract. It is not necessary that you have so much difference, huge difference. It is 3 times difference. Weight of extract I'm saying. The extract I'm not saying Lovastatin. Let us
Okay, anyways.
So is it normalized in terms of like volume?
Both be good, huh?
Okay, sir.
We'll ask him
You are
about
saying does it normalize? What is done in, okay, let me explain the procedure. The procedure is with this or
Musa, finish your presentation.
Okay, see time come. Good. Okay. After the okay, these are the different weights of the secondary metabolites collected from each from Aspergillus series exposed to different azocytidine concentration.
You're talking about the weight of cells, or you're talking about the weight of
Just the secondary metabolites. Just the second.
That's
Okay. Okay. These secondary metabolite extracts were then made into smaller solutions and then were analyzed via TLC. We used a reference of Lovastatin here, which was 1 mg per ml, and we used the running solution of ACN to acetylnitrite to benzene in the ratio of 8:2. After the run, we did the charring using barium sulfate solution, and we obtained these pink spots, which have the same RF value, confirming the presence of Lovastatin in each of these extracts. Okay. Each of these extracts were then subjected to HPLC, and the HPLC results gave a peak for Lovastatin at 7.5 minutes of retention time. We have the highest peak in A50, and similar peaks in A5 and A10, and a pretty normal peak here in the indigenous strain. The data from HPLC gave us the concentration of Lovastatin in micrograms per 100 microliter, which was then correlated to the weights of the extracts obtained and the weight and the volume of media used. And we obtained this data. We had 7.33 mg per liter of Lovastatin in the indigenous strain and the highest in the A50. The bar chart here gives the representation of Lovastatin. We have the highest Lovastatin in A50, similar in A5 and A10, and minimum in the GC17, that is the indigenous strain. Since we were also interested in studying the expression of Lovastatin biosynthesis, contrary to our expectations that with exposure to azocytidine, all the selected genes expression would increase in these strains A50, A10, and A5. It was observed that there was a varied expression. We had all the genes going up in A5, all the genes expressed, the selected genes going down in A10, and Lovastatin A and B going over increased expression in A50, while there was no change in Lovastatin F gene, and LAE showed a decrease in expression. Since Aspergillus is an endophyte and contributes to the biocontrol and the plant growth promotion, a variety of assays were done. Here is a slide showing the silver-4 production assay, that is the ability of this fungus to chelate an iron molecule. So these properties, whether it was iron chelating property, amylase property, protease.
Iron molecule नहीं होता है.
Sir,
Iron. Ferric iron.
So mistake.
नहीं ये तो ओके के लिए होता है ना. आप यही बोलना है वो.
Protease assay, lipase assay, and cellulase assay. All of these A50, A10, A5 confirm that there was no change in these biochemical properties of the endophytes after their exposure to azacitidine at different concentrations. Here are some conclusions drawn. Azacitidine treatment leads to production of new and varied amounts of metabolites, as was confirmed in the TLC. The different colors and different heights of those colors corresponded to different compounds. Azacitidine leads to increased production of lovastatin, as was confirmed by HPLC. High amounts of azacitidine treatment leads to decreased growth rate, as was confirmed by the data given by the doctor. And in the RT-PCR, we concluded that high production of lovastatin Aspergillus cannot be directly correlated to increased expression of these selected genes, lovastatin A, B, F, and LAE. As there are numerous genes located both upstream and downstream on the PKS gene cluster, which might have a superior and more central role in lovastatin production. Lastly, azacitidine treatment does not alter the endophytic biochemical profile of the fungus Aspergillus tea. Thank you.
This is the last